Good afternoon, everyone, and thank you all for joining us today. A reminder that our webinars are CPD accredited, and you will find the register in the chat, which you can complete. So this is a special edition webinar with World TB Day around the corner, and we are delighted to have Prof Robin Wood with us. Emeritus Professor of Medicine at University of Cape Town and head of the Aerobiology Research Center of the Desmond Tutu Health Foundation in Cape Town, he is a registered infectious disease specialist physician with wide international clinical experience and has been involved in HIV and TB research over the last 30 years. He developed the research platforms of the Cape Town AIDS cohort, the Gugulethu HIV treatment cohort, and the Masiphumelele community cohort. In 2003 to 2006, he was the Cape Town PI of CIPRA SA, an NIH-funded project scaling up art in Masiphumelele and evaluating the impact of art on the community TB burden. Subsequent research focused on the modeling of the HIV and TB co-epidemics in Cape Town and understanding of the drivers of the extremely high rates of TB transmission among HIV non-infected and HIV-infected populations in the townships of Cape Town. In 2013, he became PI of a multidisciplinary team of UCT scientists in an MRCSA-funded flagship project studying environmental, host, and pathogen contributions to TB transmission. In 2014, an innovative approach for isolating MTB from bioaerosols attracted supplementary funding from BMGF to further develop the approach to monitor subjects in high TB risk cohorts. More recently, his sampling and detection methodology has identified high rates of MTB carriage in both symptomatic and healthy residents of a high TB-burdened community. Today, Prof. Wood will be taking us through new paradigms in TB. Thank you so much for joining us, Prof, and over to you.
Thank you, Fazana. Let me just try and share. So screen. Can you see that?
Yes, we can. It's just not in presentation mode.
Ah, why is that? Let me have a look and see. Can anybody?
It's showing in presentation mode on our end.
Good. I don't know how to make it any more presentation than what I clicked.
On the top left, on the top left, you can swap views. Yeah, the middle one, the third one. Yeah.
Swap displays. Okay.
There we go.
Perfect. Perfect.
Okay, got you. Okay, thanks very much for this opportunity, and good afternoon, everybody. It's just turned afternoon. I'd like to present some data which may challenge the current paradigms in tuberculosis, and there is a sort of enigmatic subtitle, Hunting the Boson and Finding a Bummock, which I hope will become clearer as we move forward. So what I'm going to just touch on is a little description of the historic epidemiology of TB in Cape Town, explain what we mean by hunting the boson and what we meant by finding the bummock, and then just finish up with a possible way forward that may help implement the new paradigm. So start off where we are at the moment, and this is South Africa. As you can see, has an amazing burden of tuberculosis. Cape Town, a single city, has more tuberculosis than the United States of America, United Kingdom, France, and Canada put together. So the question is, how did we get here? And that will be the next few slides. So the first thing that I just want to highlight is that Cape Town implemented all the advances in TB control over the years, and all of them were done very timelessly, except perhaps the public sector antiretroviral therapy, which was perhaps delayed. But I'd like to highlight the bottom left-hand is my pointer showing? In 1904, tuberculosis notification of TB cases and deaths was made compulsory with a high fine to doctors if they didn't notify. Now, that means that we were about the second city, I think, in the world to compulsory notify TB, the first one being New York, and then the United Kingdom brought in compulsory notification 8 years after Cape Town. So we have an opportunity of perhaps the only place in a currently high-burdened setting where we have historical data going back over 100 years. And this was a paper that Sabine Hermans put out a few years ago. It was looking at 100 years of the differential impact of control interventions. So for all these slides, you'll see that the red line is Cape Town, the New York is in green, and the London is in blue. So the top left-hand panel, you can see there the mortality rate, a population mortality rate of tuberculosis. And you can see that 1910 to 1914, even then, there was a higher mortality at a population level in Cape Town than elsewhere, and that remained high until tuberculosis therapy came in the 1950s. In contrast, you can see that London and New York had a massive drop in mortality way before any treatment came in. So then we get to the panel just beneath there, and you can see here the case fatality. So this is people who are identified as having tuberculosis. What was their mortality rate? And you can see it was pretty much the same in all three cities until 1950. And then when therapy came in, case fatality dropped even more so in Cape Town than the other two cities. And that remained very low case fatality relatively until the HIV epidemic, which is the little perturbation at the right-hand side of the red line. If we now put our attention onto TB notification rates, which is the top right-hand panel, you can see essentially the TB notification rates didn't really change in Cape Town over that 100 years until the HIV epidemic, and then they got worse. In contrast, you can see a massive drop in notifications in London and New York, and you can see during the war years there was a slight increase in the London, but basically most of the decrease in notifications occurred in the Northern Hemisphere before the onset of TB therapy. Now, TB notifications are a pretty good measure of infection rates, although not ideal. But if you move down to the panel just below that, you look at the notification rates for under 4-year-olds. And that is much more strongly associated with transmission, because this is obviously the first time that children are exposed. And you can see, interesting, this is a log scale, but there's a log linear straight line graph in New York and London, which doesn't seem to be impacted by TB treatment. Whereas in Cape Town, you can see that the notification rates for children remained essentially unchanged. So just to the last slide on this epidemiological information, if you look at the age distribution in Cape Town of TB over that best part of 100 years, you can see that it really didn't change very much. There's a slight increase, interestingly, in recent years because of the HIV epidemic, but essentially it remained the same pattern all the way through that time. In contrast, if you look at New York, TB became a disease of old people. Children disappeared, and you can see the graphs. There is adult TB at a very low level, 2 logs lower than we have, but childhood TB almost disappeared. The last slide on the right-hand side is another way of analyzing risk, and that's looking at age cohorts. And here you can see that from 1940 to 1980, these are the cohorts at birth. So we're looking for the 1980, 40 years later, so they've reached the age of 40, you can see there's no evidence that the age cohort risk has decreased over that period of time. So, how do we interpret that? So I think there's three possible transmission hypotheses. The one which is the one that's has been the conventional approach for most of the century is all you have to do is find the symptomatic sputum positive cases and treat them properly, and the epidemic will come under control. And that's really the WHO model, Chris Dye's treatment model. But there's a problem that in currently high burden settings, you can only link a chain of infection from a known sputum positive case to another one in less than 30% of the cases. So seems to be a large proportion of people that are missed by this approach. So it could be that these people were unrecognized earlier, or that there's other unidentified sputum positive cases. And then we move on to the second hypothesis, which is really the current feeling that all we have to do is let's find these people who are sick, well, sputum positive, but for some reason haven't presented. So let's do active case finding. And I think that's probably the main focus at the moment. I did look at Cape Town, and interestingly, they had a pretty effective mass miniature chest X-ray program. At the time when the city was around about 1 million, they did 5 million chest X-rays. So it's had a high penetration. And you'll see that in the earlier graphs, there was an improvement with that strategy to some degree, but then it was discontinued. So so you can expect active case finding to have some impact, but it didn't certainly make everything disappear. And the third one is, could there be a previously unrecognized reservoir? And this could be people who are constantly in an infective state, or they could be people who are cycling in and out of infectivity. This is the Dye model of TB, and that's where I have suggested that perhaps this is hunting the boson. So the current strategies for finding TB were based on sputum and culture, and can we use modern technology to see if there is a potentially infectious reservoir that we hadn't previously identified. So there's where the Higgs boson comes in. This was a particle which was theoretically, but a fundamental component that was required for the standard model of physics, particle physics. But in fact, it was searched for 40 years before it was eventually evidence for it was found in 2012. Hence the subtitle "Hunting the Boson," that we think that it should exist, but there was no evidence for it. So again, this is analogous to looking for a needle in a haystack, and you can see that we're looking for Mycobacterium tuberculosis, typically a couple of microns long, 1/10 of a micron wide, and that multiple of these can fit onto the tip of a needle. So how do we go about finding those? Well, this was the first evidence that we had. This is a paper we published in 2018. And what we did in this one is we impacted exhaled breath onto aluminium foils. And then we subjected those aluminium foils to electron microscopy. And you can see on the right-hand panel a lot of splats, which we think are particles that are produced by the lung. Lung, in fact, is a pretty efficient particle producer as airways burst open and they're covered in surfactant or detergent, small particles are created. And when we were scanning through these electron micrographs, we came across this organism which looked remarkably like tuberculosis. It was the right shape and size. And Digby Warner, who I work with in the University of Cape Town microbiology, said, this looks like the poster boy of MTB. So it was suggestive that there might be organisms in the expired air. So we went on then to see if we could reproduce this in sputum-positive individuals. And this was the device that we developed. We call it the respiratory aerosol sampling chamber. Essentially, it's a personalized clean room. I'll go through the details of that. Essentially, if you use computational fluid dynamic modeling, you can work out what speed air currents you require in order to capture explosive events such as coughs, sneezes, or forced expiration. So we take those air streams and we monitor them. We can look at carbon dioxide levels, which gives us an idea of the volume that people are exhaling. We can look at particle quantitation, and we can collect the organisms themselves and subject them to imaging. So this is 10 force vital capacity breaths. The bottom panel in blue, you can see how each time the CO2 increases as breath is exhaled. And you can see in the top panel the particles and the particle sizes that are exhaled. And in the bit we're interested in, which is from the orange to the purple, those are from 2 microns, 1 to 2 microns to 5 microns. Those are the sort of size particles that would be required to contain MTB organisms and be able to go into the lung and remain there. So we think those are the most relevant. These are a couple of other pictures of the RASC chamber. So in the center is a liquid cyclone collecting. On the right-hand side is a high flow particle counter. And on the left-hand side are the monitoring of the environment that the patient is in. Patient sits inside, wears a Tyvek suit, and puts their head into the airflows, and all the exhaled air is captured and put onto one of these liquid wall cyclones. So we think that we've increased the sensitivity of collection, but the next big important component is the sensitivity of detection. So we combined this with this probe, which allows the rapid detection of Mycobacterium tuberculosis. It's a sugar which is taken in, it's a trehalose analog taken in by the organism, which is then metabolized, and the fluorescent component is incorporated into the mycomembrane. So that's the dye or the probe is not 100% sensitive for MTB. So other mycobacteria also contain this antigen-85 metabolic activity, and a few other organisms, most of which are not in the respiratory tract, but Corynebacteria could be a potential source of similar take-up of the dye. On the right-hand side, you see why it's called salvatochromic. It means that the fluorescence is greatly increased in the absence of an aqueous phase. So this makes it ideal for showing fluorescence in the lipid envelope of MTB organism. So what we do is we spin down and get a pellet from the collected sample, and we spread it over a series of several thousand nanowells, very small wells, whereby an organism, MTB organism, can sit in a well, but most of the noise that would distract us on the signal-to-noise ratio gets split into thousands of other small wells. So it increases the sensitivity of detection. And this is the sort of thing that we can find. You can see on the left-hand panel, the dye is taken up by rapidly metabolizing organisms, and it's concentrated in these organisms in the polar regions. And you can see on the right-hand side, you can measure the fluorescence along each organism. Most of the organisms in this phase are highly fluorescent at the poles, but you can see some with fluorescence in the middle, and those are organisms that are approaching division. This allows us to look at the length of organisms, the width of organisms, and the shape of organisms. So we can describe the phenotype of the organisms in the exhaled air. And these are the type of variety of phenotypes that we can find: long, thin ones at the bottom right-hand side, some bent ones, some nodular type patterns in group A, curved, etc., and some with relatively diffuse uptake of the probe. So we've got a sensitive, an efficient collection device, and we've got a sensitive detection device. So then we moved into what can we find in clinical individuals. And this was the first study we did. I get used to calling everything A, B, and C, but just to take you through it, group A. So patients go into the local TB clinic, and they basically get triaged or divided into 3 mutually exclusive groups. The first group are sputum positive. This is A, sputum positive TB cases that are identified, gene expert or culture positive, and they're put onto treatment. Group B is culture negative TB. So some individuals come in and their sputum is negative, but they're still defined as having TB and started on therapy. You'll notice on the right-hand thing, these are overrepresented as having HIV. So these are patients who are coming in with HIV. The treating doctor is concerned about them enough to say, I think this is tuberculosis, and start them on treatment. And group C are the rest, the people who would normally be discharged and said, we can't find TB, please look for other conditions, or come back if the symptoms recur. So those are the 3 mutually exclusive groups. And this is the baseline data that we found from them. And this was really surprising to us. The study was initially designed to see, could we find organisms in the sputum negative TB cases. But here you can see that all 3 groups have similar numbers of bacterial counts in their exhaled breath, which surprised us. And on the right-hand side, you can see the proportion of them that are positive are also very similar between the 3 groups. So now we had a little bit of an ethical issue in that groups A and B, the sputum positive and sputum negative TB, go onto therapy, but we seem to have found organisms in people who were not thought to have disease and were not being started on therapy. So we were aware that this was not a registered diagnostic. So we negotiated with the ethics committee and said, well, let's just follow them exactly the same as we follow the other groups for the 6 months. And if anything should happen, and we'll reappraise them, check their sputums and see if they move back into one of the treatment groups. And this is what happens. So you can see here the 3 groups. The purple is the sputum positive. The teal is sputum negative TB, and the ones that were going to be discharged but were now being followed up. You can see that at baseline, visit 2 is at 2 weeks, visit 3 is at 2 months, and visit 4 is at 6 months. Again, we were gobsmacked. They all behaved exactly the same. So the carriage rate went down and the numbers of organisms went down equally. The 2 groups, the first 2 groups, whilst being treated with full combination therapy, and the last group, presumably because of some immune clearance of some form. During these period, all 3 groups, their symptoms resolved. So it could be that there's an immunological clearance here, which would be fascinating to reproduce in some vaccine study, because that would be targeting transmission for the first time. So, being as we found tuberculosis organisms in all the people going to the majority of patients going to the clinics, we wondered, well, what's happening in the community from which they come and to which they return? And this is a study, again, published by Ryan Dinkoli recently in eScience. And if you look at the top panels, particularly on the left-hand side, you can see that these individuals who are randomly selected healthy people from the community, that they had very low numbers of organisms which remained pretty stable. There were a few that increased and a few that decreased with the other patterns along the top panel. And you can see again, these visits are at 0, 2 weeks and 2 months, that the proportion of positivity was much the same. And then if you move over to the scale density, it also looks as though there's no real difference between the 3 visits. If we put all that data together, so here we have the healthy controls on the right and the TB clinic patients, sputum positive, sputum negative, and suspected TB, you can see that they do behave differently. The healthy controls, the visit numbers and proportions of organisms do not change. Whereas in the first 3 panels, those that had symptoms and presented to the clinic, all of them cleared. The purples the first visit, the teals the second, and the yellows third, and they moved and cleared their organisms to slightly lower levels than were present in the community healthy controls. So this provoked this commentary in Proceedings in National Academies of Science, US, by Sarah Fortune, who introduced me to the concept of a Bummock, which I'd never come across before. And she said that perhaps we're finding this unrecognized large group of potentially transmitting, and that this could have serious consequences for the control of tuberculosis. So here we have the hummock, the top part of a iceberg, and the Bummock, the unseen part that sank the Titanic. And what we're saying is we started searching for the boson, but we seem as though we might have found something of very large magnitude that constituted the Bummock. So this is a model of our community. So you have bioaerosol negative individuals, bioaerosol positive. There's some interaction between those that we could measure from those graphs of clearance. And we have the people in the TB clinics who either have symptoms and inflammation, etc. But what we're suggesting is that people who have bioaerosol positivity, whether they're clinical or non-clinical, they have a potential for transmitting tuberculosis, and this needs to be worked and studied further. But this is really the Bummock. So we've used very technical methodologies to find these organisms, and yet we're saying that these patients who are exhaling these organisms may be in our communities, and this technology doesn't really lend itself to being deployed, particularly into low-resourced areas. I haven't mentioned the complexity that if we're using microscopy and small numbers of organisms are enough to identify the presence of MTB, then you have to have incredibly efficient sterilization processes, and we use very high dose ozone after every patient, and we do sampling of the booths before any patient is introduced into the booth, and we have consistently had negative controls for all these over the last few years. So I tried to think of some way that we could search for these organisms, and we came up with, well, let's have a relook at exhaled breath condensate. This is taking the air that's expired, you see on the left-hand side, alveolar ventilation, is 100% humidity, 37 degrees. If you were to cool that air down, you can collect the fluid. Current exhale breath condensate machines are really designed for identification of volatile organic compounds, not for particles. And it's the organisms are in the particles. So we designed a machine that we thought could collect the particles and the condensate. And this is what it looks like. It's a small desktop, and exhale breath goes into the machine, straight into a test tube. So everything that the patient is in contact with is disposable. And this shows the time temperature curve of the device on the right-hand side. So these are some of the pictures that we get from exhale breath condensate. The one on the left is a nice high definition. We upgraded our cameras to get better resolution. So that's a really nice picture of a single Mycobacterium tuberculosis. And again, you see on the right-hand side, similar phenotypic variations, similar to what we were finding in the RASC. So we can identify organisms in the exhale breath condensates. And this is just showing that we can use the fluorescence of the organism on a copper grid in order to target electron microscopy of the organism. And on the right-hand side, you can see transmission electron micrograph of a DMN tray positive stained organism. We're continuing to work on this using cryo-electron microscopy to look particularly at the cell wall structures of these airborne organisms, which we think differ somewhat from cultured organisms. And then just to finish up, I've been intrigued by lipo-arabinomannos, the test that's used for the urine identifying TB in very advanced HIV infected individuals with very low CD4 counts. And I was perplexed in that I've never really believed the story that this stuff just floated around the body and came out in the kidneys. I've always been a protagonist for saying it's a way of identifying disseminated TB and particularly renal TB. But here we looked at the exhale breath condensates of 200 patients, and we found again that we could get about a 22% positivity rate using the lateral flow assay that used to be the ALEA. And but essentially it was the same in the healthy volunteers, which are E, the patients that turned up at the clinic and were not diagnosed with TB were discharged, and the patients with sputum positive TB in group A. So there's no statistical difference between those, which surprised us. But then when we looked at the magnitude, the test has a single positive, double positive, triple positive, and a 4 positive. We did some experiments to show that represents a 2 to 3 log variation in concentrations from 500 picograms per mil up to microgram condos. And what we found is that in the healthy volunteers, that the level of LAM was much lower, and in the symptomatic patients, much higher. And if we look at the LAM, we calculate that there is 100,000 to 1 million times more LAM than is required by the organisms for structural components. So that suggests that instead of being a diagnostic, that in fact this is some sort of communication between the host and the organism, and a lot of energy of the organism is spent producing this chemical, which was known to be active, and it introduces the hypothesis that perhaps there is some control of the immune system by this organism, and that might be why we're seeing such high proportions. So in finale, I think that what I'd like to suggest is that we think that the linkage, the necessary linkage of transmission of TB to disease may not be the whole answer. That in fact there may be asymptomatic shedding of the organism and the epidemic maintained to some degree by these individuals. And this is typical of most diseases. We have stories of typhoid Mary, who's not sick but transmits the disease. Polio organisms can be excreted. Clostridium difficile can be. So many diseases. It's not, and in COVID itself, it's not only symptomatic patients that transmit. So we're saying that perhaps tuberculosis could be rethought in this sort of paradigm. So I hope I've given enough to challenge some of the current views, but this sort of work does require a lot of colleagues, and this is just to acknowledge those and to acknowledge some of the funders that have helped us with this work. So with that, I thank you for your attention. Oops.
Thank you very much, Prof, for that enlightening presentation. Please raise your hand if you have a question, or post your question in the Q&A. I'd like to just check if there are any questions from the Secretariat.
Thanks, Farzana, and thanks, Prof, for the very provocative title and equally engaging presentation. Just to kick off the questions, I'm just trying to understand the clinical implications of everything that you presented. So the data from the bioaerosol sampling showed similar levels of MTB detection in the sputum positive, the sputum negative, and the unknown, in terms of the individuals that had tested. So how should these findings influence, the clinical management as well as infection control strategies in the healthcare settings as well as in communities?
So the last slide, which I didn't show you, was the dye model, which they postulated of individuals moving into an infectious phase and moving out. And I think that's the group C that we're seeing. And what we're saying, for a limited period of time, those individuals that have come to the clinic may be exhaling potentially transmissible organisms, but they do clear themselves with time. So we spend a lot of time going out into the community to find people who may be be sputum positive but not symptomatic. But we are getting people who are symptomatic that are coming to the clinics in large numbers, much greater numbers than the ones we treat, and we send them away. They may well be transmitting during that period. So that's the first thing. So if you're modeling transmission studies, you could take those into account. The second one is that I'm intrigued by this spontaneous clearance of organisms to very low levels by the group Cs. And that would suggest that there is an immunological response which is clearing the organism. If we could reproduce that in a vaccine, then you really truly would have a vaccine targeting transmission and decreasing aerosolization of organisms. And then the last one is just separating transmission being only amongst symptomatic individuals. But the reason that we try to change the technology to make something that's a little bit easier to find is that we don't have the technology for diagnosing these aerosols outside of these sophisticated laboratories. And that's why I think that I'm hoping that EBCs, which are very exhaled breath condensates, which are very easy to collect, may give us an idea of how we would find these individuals. But this is very early data. The fact that I found organisms doesn't necessarily confirm that they are transmitting, so we do need to do transmission studies. Yeah, so it's really changing the paradigm I don't know how this is immediately going to change the control system.
Thank you.
Thank you. Thank you for that, Prof. So we do have a number of questions in the chat, the Q&A, and I think let's start with Janet. Janet, go ahead, please. You can unmute and ask your question.
Thanks. Thanks for the chance to ask a question. I have posted in the chat, but maybe I'll explain it a bit more. Robin, thanks for an excellent, very interesting presentation. I suppose so my question is what you are demonstrating in this, let me call them the group C individuals, is this TB infection as we understand it in that spectrum? And the question being, how transmissible are people with TB infection as opposed to, say, asymptomatic TB, but I know it's a spectrum. But I think my main question is, how should, if you did have an easier, a more sort of feasible diagnostic test for these individuals, I see somebody's asked a question, is there a correlation with the IGRA status? But how should they be treated? Should they be treated with a full course of TB treatment, or with what we now call TPT, which, that policy says it's the treatment of TB infection. So it's not really preventative. And it's always confusing, I know, when I teach about this, is this preventative treatment or is it treatment? It's actually treatment of TB infection. And would that be sufficient? And as we currently are trying to increase the uptake of TPT in close contact, would these be the individuals who would be benefiting or not benefiting from TPT? So it's really all about the treatment and TPT and what's happening currently.
Yes, so I would suggest there's probably three states. It's possible that this is a colonization state. There has been some work in, I think in Nature, of lung tissue showing that cords of MTB can survive on alveolar cells, and then they could be shed. And that might be what we're finding. There's infection, and then there's disease. So disease is obviously you require end organ damage, and that's why sputum is such a good measure for that, for disease, in that it's showing inflammatory cells in there. Infection is a little bit more of a problem in that we don't find a correlation with IGRA status. So that's the first thing. Second one is, I'm not a great fan of the terminology of prevention. And my feeling is that if you've got recently infected, your risk of progressing to disease is higher, and therefore IPT is in fact a little bit of treatment for a little bit of disease. I did touch on to the structural components of these. When we look at the culturability, we think that the reproductive time is fairly slow, a few days rather than hours. Interestingly, these organisms, as has been recognized before, are in fact ZN negative, and this has been recognized for a long period of time. So that's why we're looking at the cell walls. And obviously, if the cell wall is somewhat different, then the conventional prophylactic regimens are targeting at cell wall components. So that's why I think the structural component is really quite important. But being as these patients actually clear within 6 months the group Cs on their own and go to lower levels than are in the community from which they come, I don't really see there's a need to treat them. It may be interesting to look at, are they spreading to other individuals? And I think what this sort of work also helps is, whereas the chains of infection are defined at the moment from one sputum positive to another sputum positive case, if we can improve the technology so that we can barcode these organisms or identify SNPs in them that can link chains of infection, it will be a great way of better understanding the epidemiology of transmission rather than just looking at sputum positive to sputum positive, which is only explaining a third or less of the transmission. Does that actually answer your questions, or have I waffled onto some other topics?
Well, I think there's obviously a lot of unknowns here. I think this whole question of asymptomatic TB as well is difficult to know. And I mean, when you say the cell wall structure is not, you can't see Nielsen stain it. I mean, what is it? Is this a kind of inferior, I don't know, alternate form of the bacteria? Is it? I don't know enough about that to understand. But
okay, well,
but yeah.
So this is not new. 100 years ago, these organisms had been identified in tissues, and they were ZN negative. They were injected into the peritoneum of guinea pigs. They killed the guinea pigs with TB, and the organism changed back to conventional ZN positivity. So, yeah, so there's a lot of different phenotypes which have been recognized for a long period of time. You're right. We need to show transmission to people within the same household of these, but the problem is we're dealing with so few organisms. It's very difficult to get whole genome sequencing data. We have sequenced some of the group Cs and shown that that they are MTB, but you need 1,000 organisms in order to get a whole genome sequence from this. But technology is changing all the time. If we could get down to a handful of organisms, then we'd be able to identify these specific organisms that we're finding and see what the transmission patterns are. The other way is to look at things like RNA and RS ratios, where we know there's much more RNA than there is DNA. So the problems we have with this organism is that we're dealing with a handful of organisms, and it's basically a ball of wax with a inefficient way of exposing the DNA, whereby you lose half a log to 1 log. So you can see that if you start off with 20 organisms, you're down to 1 or 2 by the time you've copies of DNA. So the whole thing is challenging technologies, but the technologies are changing rapidly. So I think watch this space.
Great, thank you. For the next question, I'll take one from the Q&A box from Lucy. She says, interesting data, a few questions. Were the healthy controls contacts of the clinical attendees, and did you screen for infection in these controls? You mentioned the three groups of clinical attendees all had MTB carriage that declined in a somewhat similar trend over the follow-up period. Were there X-ray results for Group C, and what proportion of these were later diagnosed with TB?
There's a few questions there. The first one is the controls were randomly selected from a map of ERFs in the area, and then everybody in the randomly selected number of ERF was tested. So there was no connection with the cases that in the clinic. So they were apparently healthy individuals. We did not do chest X-rays on them, but they were asymptomatic and they were randomly chosen. Future studies, we will be looking at those. And the Group Cs, again, we're trying to do a study whereby we're looking at defining them a little bit more precisely by saying that they are presenting with symptoms but don't have chest X-ray evidence of inflammation. We're looking for other markers of inflammation as well. We can look at protease activity in the EBCs. We can sometimes find that, which shows that there is some inflammatory process going on. We're looking for metallo proteases, and we've used mass spectrometry to look at this. So there's a lot of other data coming in, but all of it is somewhat high-tech stuff to try and answer these questions. But my feeling is that we were as puzzled as everybody else when we saw the data for Group C, and it's really been intriguing and intrigued a lot of people. And where it will go, we're still trying to find out. We need to prove that they can transmit, and that would require looking in households with exhaled breath condensates, for instance, but it's still the problem of we're only dealing with very small numbers of organisms. And how can I say that my organism is the same as your organism if you just transmitted it to me recently, and neither of us have sputum positivity? That's the technical challenge of porcibacillary bacteriology, which I think is a field which has got to grow in the near future.
Okay, next question is from Rubeshan. Thanks for the excellent talk. In comparison to the bioaerosol, do you know what proportion of asymptomatic TB cyclical shedders would be detected by currently available molecular assays or by culture?
So the group Cs are not asymptomatic. They came to the clinic with symptoms compatible with TB. We realize that we now need to screen them for other respiratory pathogens. We know there's an annual cycle of TB with peaks at certain times of the year. People have argued that that might be related to the cold weather crowding, could be related to vitamin D status, or it could be related to co-infections with other organisms. So we've only been dealing in the clinic with symptomatic, so I can't really say how good this would be at finding asymptomatics. During our random selection, we found, I think, just under 1% of patients that actually did have sputum positive TB, and that fits with lots of other surveys that people have looked at and we've done in the previously. But we haven't really looked specifically at how do you find asymptomatic individuals in the community, except for these very low numbers that we're finding. I know there's a big study, Gates-funded study with thousands of screening, looking at that in Indonesia and in KZN, but we don't have results that really inform that.
That leads us to the next question, which is, could the data suggest that asymptomatic young children may also be transmitters? And did these potential transmission dynamics not apply in London and New York due to less overall burden of disease?
So I think that's a really fascinating question, because my heart of hearts, I think the answer to all our control programs is what's happening to children. And part of the reason that we developed this exhaled breath condensate is that for the first time, I think that young children can blow into straws as well as anybody else. So it gives us the opportunity to look there. Then the next part of that question is, could children be transmitters? And the standard feeling is no, because they don't produce sputum. But if we're separating transmission from disease, then I think it needs to be rethought of, and it could do. And we have the problem that up to the age of 14, transmission is taking place at a very high rate, but disease is at a low rate. So it's certainly a possibility, and we need to see whether that is the case. But again, we're dealing with so few organisms that we need to have the technology to be able to identify and get a specific strain pattern from these very small numbers. But it's a question which is in my mind. The question is put forward, and I think it's certainly possible, but I can't say that they are transmitting at the moment.
Okay, I think let's take Tom Boyles. Please unmute and raise your hand. Unmute and ask your question.
Thanks very much. Thanks, Robin. I find it absolutely fascinating. Yeah, I was sort of intrigued by these group Cs.
Everybody is.
Yeah. I've always felt that, we've lived with TB for millennia and that we haven't learned to live with it, because it needs to make you sick to transmit well. And that's why it's not CMV or something that we just live with. It has to make you sick, but it doesn't want to kill you. in terms of these group Cs, I mean, they're all sick with something. Is it not a case of TB kind of hijacking this inflammatory response to somehow evade the immune system? I mean, we know that people with pneumonia and other things, they cough up dead bacilli. With something as blunt as a gene expert, we pick that up. And so I was just intrigued by you saying about a vaccine for these group Cs. Is it not more that this is a natural perturbation of the immune system by the thing that they're sick with, which somehow TB potentially seems to be using to its advantage? Again, having to sort of coexist with sickness to become a more efficient transmitter. That's all I was thinking.
Okay, so the first part of your comments and question was about the sort of evolutionary interface between Homo sapiens and the organism, and I think that's a really great way of thinking about it, in that this organism has been with us for tens of thousands of years, and it's not in its interest to cause disease. It doesn't, the organism doesn't produce anything really in the way of toxins or anything like that. It just produces an immune response, which can, under certain circumstances, cause quite a lot of damage, TNF alpha blockage, etc. So I think the evolutionary view, what would the organism want to do? And you think, well, it wants to be everywhere but not cause disease, so that it can spread to the young of the next generation, etc. So I think that's a really important way of looking at it. The immune response of the group Cs is also fascinating. Is this a little bit of recognition of the immune system of this organism, and somehow or other its head is poked above the parapet and it needs to go down and get controlled? We're in sort of area of supposition, but I sort of think that may be the case, and it fits in with the sort of dye model of alternating infection and clearance. The reason for the immune response was, as you were quite right, the Cs are doing it all on their own, but if you could give it to the rest of the community that cleared their exhalation of organisms, that would be targeting transmission. So that's really where I was thinking. And then the last one is the LAM. LAM is much more complex than people think, in that it's like a tree with leaves, a trunk and roots, the lipid anchor, and we recognize it only by the leaves. We don't actually know whether it's AM without the lipo, or whether there's different structures to it. Certainly we find that mass spectrometry is very difficult because the molecular weight is so variable, and therefore the charge, and you can't identify peaks. I and many of my staff went 4+ positive on LAM for a few months last year, and now are all clear. And that's why I was intimating that, is this a way the organism actually talks to the host? Why would it evolutionarily spend so much metabolic activity producing something to 100,000 times or 1 million times what it requires structurally? So lots of fascinating questions, but I do like your approach to thinking of it in the long-term evolutionary relationship that we've had with this organism. I probably haven't answered any of your questions, but
no, you have. Thank you. Yes, I mean, I'm a evolutionary biologist at heart, so I always think of things like this. I mean, we should have learned to live with this organism the way we've learned to live with CMV, for example, and probably would with HIV over time, because it's not in their interest to kill anybody, but it seems to have to make you a little bit sick to transmit efficiently. But what you're suggesting, maybe it doesn't have to make you that sick, actually.
So I'm not
to
sure
transmit a bit.
So it is fascinating from an evolutionary point of view. The trouble is that all our technology was aimed at culturable organisms and selecting organisms that grew well in conventional media that we sort of experimentally produced. These organisms, what I've done is try to explore their reproductive capacity, and some of them we can get up to 1,000 organisms, but many of them are much lower than that. And what we do is we do a sort of DNA count at day 0 and using RD9 specific for MTB, and then looking at that after 50 days of culture. And you see increase in the number of DNA copies over that time, but nothing like the sort of 50 to the power 2 to the power 50 that you get from an organism multiplying every 24 hours. And that's where I get the calculations of the relatively slow multiplication. But the fact that we have concentrated on organisms that specifically well adapted to our growth media doesn't mean that there are other forms of organisms.
Agreed. Thanks very much. Appreciate it.
Thank you. There's just a couple more questions in the chat, if we have an extra few more minutes. So I'll just read them all out. Is there a correlation between IGRA status and count of particles from respiratory chambers? Are there any male and female differences? And please explain the ULAM concept again, which I think you may have just done already.
So the IGRA positivity rates didn't correlate with the number of organisms. And I think the reason for that is that when we put this into a model, it seems as though the exposures are several times a year. So my feeling is that the IGRA is probably more representative of the group C's and A's. So if you look at the people in the clinic, almost 100% of group A's sputum positive are IGRA positive. The rest of the people, the B's and the C's, are lower than that, and the general community is lower than that. And there is a clearance rate, so that it's really when your infection rate is higher than your resolving rate of the IGRA, it gets quite complicated. But no, there is no direct relationship with IGRA, but IGRA positivity in our communities is incredibly high. Then, interesting, we didn't find a male-female difference, but I'm not sure that we powered it particularly for that. So if we had a hypothesis that there was a difference in transmissibility, then it would need larger numbers to look at. So it's interesting. We didn't find a big difference in HIV either. Yeah, that's the negative data. But negative data always is dependent on, did we actually have the power to answer the question in the first place? And we stopped our study prematurely because we suddenly, once you've got 98% of all the 3 groups, looking for differences between them becomes a little bit academic. So the study was stopped earlier than had been planned, and the plan had been predicated on a difference between on the group C's being negative and the difference between the A's and the C's.
Thank you. Thank you, Prof. I think for the sake of time, we'll end our question and answer session there. I'd like to take another minute just to thank you, Prof Wood, for this enlightening presentation, and thank you all for your questions. Please email Nolwazi on the email address on screen if you have anything that you would like to present at our upcoming webinars. Thank you for joining, and thank you from myself, Fazana, TB Think Tank Scientists. Have a good day.
Thank you.